Okay. All right. I guess we're also live now. Let me share. Okay. So do you see the full screen slideshow or presenter view?
Yeah, I think it's full screen. It's good.
Okay, sounds great. All right. So then, hello everyone. This is the first lecture of the project and seminar course, Accelerating Genomics. And today we're going to cover the logistics and also a brief introduction to the steps, to the computation steps that we follow in genome analysis. So the real head supervisor of this course is Mohammed Alser, but unfortunately, due to some unforeseen circumstances, so he cannot be here today. So I will be providing the logistics and also giving the lecture today. But he's the actual head supervisor of this course, so you can contact him regarding the logistics or anything regarding this course throughout the semester. So, all right. So then what is basically the role of this course? Let's first cover that. So basically this is a projects and seminar course, obviously, and in accelerating genomics. And what we're gonna do, we're gonna be basically covering the basic step in genome analysis. But while doing so, you're gonna be getting familiar with the key algorithms that we usually apply for analyzing the genomic data and also to make predictions based on the genomic data. So so I'll also answer some questions. So I just saw a question in chat. I'll answer those on certain times. And while doing so, you're going to be getting familiar with the algorithms. And these algorithms, some of these algorithms apply some heuristics. Some of these algorithms are actually very optimal, and there's going to be some trade-off between those algorithms. So heuristics algorithms maybe are going to sacrifice from accuracy, but they're going to provide really fast analysis. And some of the optimal algorithms are going to be slow, but they're going to be really accurate. So there's always going to be some trade-off between those algorithms. And what we are going to do throughout this course is, as you get familiar with those algorithms, so you're going to be experimentally evaluate these different heuristics, these different algorithms, and then you're going to be observing the effects of these heuristics or applying the optimal algorithms on the end result of the genomic analysis. So what you're going to be, for example, seeing that when you apply a certain heuristic, you're going to see that the steps are really fast, but maybe it's not that accurate. And then you're going to decide that, okay, I want some really accurate analysis. I want really accurate answer from this step. So I want to apply an optimal algorithm, let's say. So you're going to be really experimenting on these different algorithms in different steps of the genomic analysis. And so as you apply these heuristics, so this is the evaluation will give you the chance to carry out a hands-on project, basically, right? So you're going to be learning from the algorithms, but also you're going to be implementing these algorithms on a hands-on project. And these heuristics that you're going to be applying are hopefully going to help the society by enabling fast and on-site analysis of genomic data. So, okay, so we have, so what are the key objectives in this course? So we really want to enable you to grow in multiple aspects. So what are these aspects? So first of all, you're gonna be hopefully improving your basic knowledge in genome analysis. So this is not, so what I mean by genome analysis, you shouldn't be imagining something on the wet lab, meaning, so you're not gonna be really dealing with the real samples, such as a blood sample or something else, but you're going to be actually dealing with the the computational problems in the genome analysis, which we call the dry lab. So you're going to get familiar with the basic knowledge in genome analysis. So this is one aspect that you're going to hopefully improve throughout this course. And another aspect is that you're going to be improving your technical skills in genome analysis and computer architecture, meaning you're going to be learning like these different algorithms, and also you're going to be learning different data structures, and also you're going to be learning like these different computer architectures and how we can really leverage like each different type of computer architecture in order to solve the computational problems in genome analysis fast and accurately. And another aspect is that you're going to be hopefully improving your critical thinking and analysis. So as you try to solve the problems, you're going to be, of course, like sometimes failing. And then as you fail, you're going to be thinking critically. And then we try to observe like what we've done wrong and then what we could have done instead. And then we're going to try to solve the problems that you face in an intelligent way, let's say. And of course, like another aspect is that you're going to be getting familiar with the key research directions, right? So you're going to be learning the state-of-the-art research in genome analysis, and then you're going to observe like what are the key directions of today's in genome analysis, what people are doing nowadays. And then as you get familiar with those, you're going to be hopefully also trying to think about like the potential solutions that we face today in genome analysis. And last but not the least is you're going to also be improving your technical presentation skills. So especially towards the end of this course, we're going to give you, let's say, strategies how to communicate, let's say, your analysis and your report clearly to your peers and also to your supervisor, so that you can clearly describe what you've done throughout this course. So basically, like these are many aspects that you're going to be hopefully improving throughout this course. And then we're going to, as your supervisors, we're going to guide you so that we can enable your growth as much as possible. So what is the key goal of this course, right? So the key goal is basically you're going to learn how to efficiently implement one of the key steps in genome analysis on a suitable hardware, right? So this hardware can be anything. It can be processing in memory, it can be GPUs, it can be SIMD processors or anything. So this is really up to your imagination. But like as we implement the algorithms, we will also want you think about the suitable hardware that you could apply your implementation on it. And then maybe you can also implement in a way that it can be also suitable for that hardware. So you're not necessarily going to implement something specifically for that hardware, but it will always be good for you if you could basically think about this suitable hardware as you implement. For example, if there are really like embarrassingly parallel steps on a problem that you're solving, maybe you can think about like how you can really solve that problem in GPUs, right? So that you could exploit these many cores that the GPUs are providing you. So so what are the prerequisites of this course? So I think we mentioned this in an email or in the course catalog, but so we don't really require any prior knowledge in bioinformatics or on genome analysis. So we're going to be really providing you the basics throughout this course so that you can get that knowledge from us. But what we require is that for most of the projects, for almost all of the projects, we're going to be requiring you to have a good knowledge in C programming language or C++ or Java, like basically any one of the high-level languages is required. And another prerequisite is, of course, like we want you to be interested in making things efficient, and also you should be really interested in solving hard problems. So yeah, because like you're gonna be having like these different methods to try, and then you're gonna fail. So as you fail, you should be really eager to try another method and then try to solve it again. So I guess like we're gonna have like around 10 weeks in this course, and during those 10 weeks, you're gonna be of course, communicating with your supervisor, and then you're going to be, I guess, developing the strategies like on each week, like maybe in earlier weeks, you're going to be just learning the topic by reading the materials, and then later weeks you're going to be implementing the project, and then last weeks maybe you're going to be doing your analysis. So your supervisor is going to help you with that. But I guess like during the last few weeks, many of you, maybe all of you, are going to have your exams. So you can develop different strategies there. So you can either stop right before your exams start, or you can also decide to continue after your exams. I guess we're gonna have these project presentations definitely after the exam period. So you'll have some time to have some progress on your project after the exams. So this really depends on your progress and like also your eagerness, let's say, if you want to continue working on the project after the exam. So this is basically pretty flexible in terms of the time management. So you basically, your supervisor is going to help you with that. Okay. So let me briefly talk about like who we are in this course. So as I said, the head supervisor is Mohammed Alser. So unfortunately, he's not here today, but he's the lecturer and also the senior researcher at ETH Zürich in the Safari Research Group. So Safari Research Group is led by Professor Onur Mutlu, and like there are a few senior researchers in the group, which you're going to meet some of them also soon, hopefully. So Mohammed got his PhD from Bilkent University in Turkey in 2018. And he worked at several places, for example, UCLA, TU Dresden, and Petronas. And then he received also a doctoral dissertation award from the IEEE a few years ago. So you can see his Twitter account there, and his main research is mainly in bioinformatics, computational genomics, metagenomics, and computer architecture. So yeah, I guess like in short, he's mostly excited about like building these new data structures or algorithms that are more intelligent and suitable for certain hardware design. And we also have Juan, who is also a senior researcher. He's really an expert in GPUs, also in processing in memory, like basically heterogeneous computing. When you think about like how you can use like multiple hardware in a heterogeneous manner. We also have Nur, who is also a senior researcher, who also could not be here today as well. He mostly works in data privacy, bioinformatics, and in general computational genomics. And also you have me, Can Firtina. I'm a PhD student in the Safari Research Group, and I'm also mostly interested in developing algorithms for efficient and accurate genome analysis, while again like thinking about like how to accelerate those algorithms. And okay, so we also have Jeremy, who is a PhD student. He works on several topics like the improving the performance and the reliability of the DRAM or the computer architectures, and also he's interested in genome sequence analysis. And we also have Joel, who recently joined us as a PhD student, but he was also previously working with us during his master's studies. So he first like started doing research with us before he joined as a PhD student, and then later he continued as a PhD student here. And he's mostly interested in accelerating the bioinformatics pipeline, right? Like multiple steps in the bioinformatics pipeline, and then he's also interested in like doing other things like other than the bioinformatics, like current and future computer architectures, and then how to improve them. So we also have master students who are Max, Banu, and Arvid. So throughout this course, they're going to be also helping you with solving the problems, and yeah, basically with any other things. So what are the course requirements and our expectations? So these are actually essentially needed for you to be successful in this course. So you shouldn't be really thinking about the grade that you're going to receive, but you should really be thinking about how you can achieve the highest growth in this course. And we really want you to help. We really want to help you enable that, basically, so that you can be successful in this course. And to achieve that, attendance is really required for all meetings. So we're going to be probably having these 2 meetings every week, but those meetings are going to be identical. So you're going to be required to attend either of these, like at least one of these lectures you should attend. And so this is not decided yet, but later on we may also decide to merge these meetings. So rather than having multiple lectures every week, maybe we can have like single lecture in a week, and then you would be required to attend that one. And another thing is that we will expect you to study the learning materials that we're going to assign you, which are usually the readings, the papers, and the videos that you need to watch. And I guess like some of the learning materials are already assigned. Hopefully you read or watch some of these the materials that you already have. And so each of you are going to carry out a hands-on project. And what this means is that you're going to be building, implementing, coding, and designing the project itself with a close engagement from your mentors, right? So they're going to guide you in a certain direction, but what we will be doing is as your mentors is that we're going to try to give you the directions, but really what you're going to do is that you're going to get your hands dirty, and then you're going to be implementing the code, and then do the analysis, and then observe. So what are the bottlenecks, or like what are the issues in your implementation or in the implementation that you're going to work on, and then you're going to discuss your observations with your mentors, so that your mentors can also continue guide you in the right direction, let's say. So for that, we really expect you to participate throughout this course, right? So participation means you should participate during these lectures, but as also you should participate during your meetings that you're going to carry out with your mentors. So you're going to have these meetings with your mentors, and then you will be expected to carry out like basically the critical thinking, or you should be asking questions or asking help basically from your supervisor, so that you can continue participate, you can continue working on your project, so that you can have a good progress on the projects that you're working on. And also you should be reading relevant papers, so that you can learn on the topic that you're working on. And mentors are going to be assigning you the papers so that you can read and learn, right? And while you're improving your or implementing your project, you're going to have a GitHub repository, which is going to be private initially, and you're going to be committing your codes there so that you can be in sync with your mentors. And later on, like throughout the end of this semester, you're going to be giving a presentation to everyone, meaning to your peers and also to your mentors, right? Professor Onur Mutlu is probably going to be there, and also maybe like all the other PhD students in the group are going to be there, so that you can present your work, and then so that we can discuss what we can do next, right, to continue solving the problems where you left off. And if you basically do a, let's say, really good job, and then if you see a good progress on your project, we may even help you to get your work get published in good venues, right? So we can help you write a paper and also get it published in good venues if this is also what you want from this course. Yeah, I think I mentioned this, but your responsibilities, as I said, you should attend either of the lectures every week. So one of them is currently scheduled on Tuesday at 10, and another one is scheduled on Friday at 9. And the lectures are going to be 1-hour lectures. So we're going to be providing, we're going to be teaching you basically different topics in genome analysis. And we're going to be also mentioning recently published papers from our group so that you can get to know also our research, right? So that you can get to know the state of the art in the research in genome analysis. So as I said, attendance is mandatory. You're going to be expected to work on your projects probably around like 6 hours per week. So this is not really strictly required, like you should work on every week 6 hours. Maybe like for some weeks you're gonna work 2 hours, another week you're gonna work, I don't know, 10 hours. So this is really flexible, but like more or less, like you can think of like every week you are expected to work for 6 hours for the project. And then you should also be meeting with your mentors weekly. These are going to be one-to-one meetings so that your mentor can provide you good guidance, let's say. This is the website of the course, so you can visit the website so because like we publish the slides there, and then we publish the learning materials there, so you can access all these materials in the website. So you should also check your email and Moodle frequently so that you can get the announcements from us. And also we're going to be assigning you some tasks, maybe homeworks, and then you can access these homeworks in Moodle, and then you can submit your report or your answers, let's say, throughout the model. So you should be checking those as well frequently. Yeah, I guess this is about the course website and Moodle. And for the next meetings, at least for the next meeting, we'll give you a chance to select your project. So after this intro part, we're going to be describing the projects that you can select. And then after we present you the projects, you're going to have the chance to select the project. So you'll have the chance to rank the projects that you want, right? So it's not just a single project, but you should really be ranking them based on how much you like them, how much you want to work on them. And then we're going to assign the projects based on your preference, but also based on your background. So in order to assign you, hopefully, the best project, we're going to see your decisions, and then we're going to have like a one-to-one meeting to match your interests, like skills and background, and then we're going to finalize the project that you're assigned on that one-to-one meeting. And this meeting is probably going to be next week, and hopefully you're going to start working on your project as well next week. So again, like it is important that you study the learning materials before our next meeting, so that you can also make a good decision on the project that you want to work on. Yeah, I guess that's all. So I guess this is all about the logistics. Do you have any questions regarding the logistics so far? So I see a question in the chat, which is just out of curiosity, are the lectures on Tuesday and Friday are identical? Yes, they are identical. And yeah, later on, we can also decide to merge them. So rather than having 2 lectures, we may have a single lecture, but we'll see. We're gonna think about this and then see what we should do best about this. Do we have more questions? Okay, I guess no questions. Then I'm gonna move on with the intro to the genome analysis. So we're done with the logistic right now. Okay, so what is genome analysis, or what is a genome, right? So here in this picture, you see multiple faces, like different characteristics, different phenotypes, different body shapes, right? So this is basically what you see different in each of these individual, like we call the phenotype of that individual, right? So everyone looks different here, right? So, but like the question is like, what causes all these differences, right? So when you think about this, so all of these differences are actually coming from from your genome, right? So genome is your individual's complete set of genetic instructions. So it gives you the instructions on like how your skin color looks like, how you behave, even how your body shape will look like, etc. So like the phenotypes and like also the other things that you're not seeing, how you're how much you are prone to certain disease, etc. Right? So these are really coming from your genome. So it's basically like almost all the answers to the questions that you may have about a sample, and this sample can be a human genome, it can be a bacteria, can be a virus, anything, right? So and like, so this is basically how it looks like. So this is how it looks like in a text domain. So a genome is really a chemical molecule, of course, right? And then we have these ATGCs in our DNA, and like, and we, in order to basically do analysis for a genome, we convert that into a text sequence, right? So that we can perform some computational analysis on that. And like these are basically the chemical molecules, as I said, the ACTGs, which are the adenine, thymine, guanine, and cytosine, right? And then this is how they look like if you convert them into a text sequence. So then what are the applications of the genome analysis? So what we can really do with the genome that we read, with the text version of it, right? So this is like, the applications are really unlimited. So this is really up to your imagination, let's say. So we can use these text sequences so that to understand like what are the genetic variations between each individual, right? So what is basically between the individuals, so that a certain individual looks different, or certain individual has like more tendency, for example, to develop a cancer, right? So this is, for example, what we can do. We can compare the text sequences. So what we can do also, we can do the prediction of the presence or relative abundance of the microbes in a sample, right? So, for example, you can get a sample from an underground, and then you can try to observe whether there are dangerous, let's say, viruses developing there, so that you can quickly decide to kill them, right? So that you can quickly decide to isolate that environment so that individuals won't get affected by that virus. So what we can also do is that we can do which is similar to this one, but we can do a rapid surveillance of the disease outbreaks, right? So similar to COVID outbreaks, researchers are continuously sequencing, reading the DNAs, and then they are trying to figure out whether that certain sample that they sequence, the certain sample that they analyze, right, are really dangerous for human health or for the environment. And another thing is that we can do or we can perform like, or develop personalized medicine for each individual, right? So not like each drug that you take are best for every individual. And what we can do is that we can analyze your DNA and then decide which sequence of drugs can be best for your condition, so that you'll get the best out of the medicine that you're going to receive. So, but like, these are only the examples that you can do when you have the DNA sample, and then but like, we have like many more examples, and then hopefully you're going to be learning about these examples later on this course. So then how do we really analyze a genome, right? So simply how it sounds like, for example, we have your blood sample, and then we send that blood sample to a lab, and then they analyze your genome, and then they'll give you the answers, right? So when you look at that, in the high level manner, this sounds really easy, right? But actually there are lots of problems between the steps where we get the sample from you, and then where we provide you the prediction. So there are a lot of computational steps going on there. There are a lot of actually also biological steps that are going on there, so that we can give you the most accurate and also the fast answer about your DNA, or about the results that you want to see, let's say. And one of the main problems is that we currently do not have any machine that gives you the complete sequence of your genome as output, right? So it cannot take for example, blood sample of your genome, and it cannot give you like this one huge sequence of your genome as output, so that we can work on them easily and then do the some analysis, right? So one example, of a company doing such analysis is the MyHeritage company. So what they are doing is that they are getting your sample and then they are providing you the analysis. So they are sending you a package, and then you are expected to ship the sample to them, or you can just give the sample to them, and then they will basically give you some answer regarding your DNA. But like what's happening is that, so what are they really doing is that they are providing your sample to the machines that we call high throughput sequencing machines. So these machines are taking your DNA sample as input, and then they are providing the text version of it, let's say text version of your DNA as output. And there are many machines doing that, right? So these machines can be really huge, or they can be really hand-sized machines. And like they have like different characteristics. Like for example, some of them are really fast, but erroneous, let's say. So they have like these different characteristics, and then the price also using them also varies based on the characteristics they have. But like what these machines are really doing is that they are choppers, basically. So what they do is they get your DNA as input, and then they actually chop your DNA into pieces. And then they provide the chopped version, the fragments of your DNA as input. So what is the problem with that, right? So the problem is that, for example, human DNA, a single, let's say, chromosome, regardless of like if it is coming from either parent of yours, 23 chromosomes actually is about like 3 billion characters, right? And these machines can usually provide somewhere between like up to few hundred characters, from few hundred characters up to millions of characters. So meaning that actually billions of the fragments are produced from these machines. And these fragments have two main problems. One of the problems is that we do not know where that fragment is coming from your genome, right? So it's just providing a fragment, it's just providing a text sequence, but we don't know where it belongs to in your genome. So one of the goals is actually to find out where it belongs to in your genome, so that we can let's say build the complete picture of your genome from the pieces. And another issue is that the machines are going to introduce their own errors in the fragments that they are producing. So we're getting some fragments, but those fragments can also be erroneous. So meaning that the characters that you see here may not really be coming from your genome, but they may be there just because the sequencing machine produce an error over there. So this sequencing machine takes the DNA as input, produces the fragments of the DNA, which we call reads, because it reads your DNA and provides the fragments. So we call them reads. But these reads are chopped, lacking the information, information of the location, and also they are erroneous. So this problem is like solving a puzzle, right? So imagine that we have a picture which is completed before, which we call a reference genome, right? For example, we have the reference human genome, which researcher completed around like 2003, and then it's getting improved every year. It's getting more accurate, etc. So we have that picture, which we call the reference genome. And the goal is that by looking at this picture is actually when you get the fragments, when you get the reads from the sequence machine, the job is by looking at that picture is to complete that picture again, right? But if the pieces are really small, then the problem of yours is really hard, right? Because like when you have the smaller pieces, it's really ambiguous where to place certain pieces on the correct location in the picture. And another thing is that those pieces are going to be erroneous. So when you place them, maybe you're placing them in an incorrect location, right? So ideally, what we want is that we want large pieces, and we want really accurate pieces. And as I said, like some sequencing machines provide like these large chunks, large reads, but they are really erroneous. And some machines provide really small reads, really short sequences, but they are really accurate. So there are really trade-offs between them. But the goal is, again, as I said, is to complete that picture by looking at the picture that we already have, which we call the reference genome. And computationally, how can you solve this problem, right? So you could maybe apply a brute force algorithm, meaning that, so assume that this is your reference genome, which is already built, which is already there. And then this is your single read that you receive from the sequencing machine. And then what you can do is that for each location, you could look at like, does it match, does it match, does it match, right? Until the end of the reference genome. But remember that this reference genome is huge. For a human genome, it is about like 3 billion characters, and your read is somewhere between like 200 characters, maybe even up to 1 million or 2 million characters. So when you do the brute force algorithm, it is really expensive, right? So it depends on the read length, it depends on the number of reads that you have, and it depends on the reference genome length. And the number of reads is usually millions to billions, and the read length, as I said, is few hundreds to millions. So it's really expensive when you think about it. So then what are the heuristics, for example, that we are applying to solve the problem that I just mentioned? So imagine that this is a piece from a reference genome, right? So this is what we call a FASTA file, and the FASTA file mainly includes the information about the sequences about your DNA sequence, right? In a human readable form, and then it usually has 2 lines per sequence. The first line is the identifier of that read, and the following line is the actual sequence of the DNA. So here, for example, this is the identifier, and this is the sequence that we have in the FASTA file. And another format is that we can have the FASTQ file. And the difference between the FASTA file and the FASTQ file is that it includes the information, the quality information of the sequence. So some of the sequencing machines will give you, let's say, its confidence about the base, about the character that it produced over there, so that you can decide to rely on that sequence, or decide to rely on that character or not, looking at the quality. But essentially they are the same, right? So it has the identifier as well. It's the sequence, so this is some auxiliary information, so you can ignore it, and then this is the quality. So assuming that this is the reference genome and this is the read that you receive from the sequencing machine, so one of the heuristics that we're applying is that what we're doing is like we're fetching only small pieces from the reference genome, right? For example, this small piece is, let's say, green, and then like this is blue, etc. But like we're fetching like fewer and smaller pieces from the reference genome. And then what we're doing is that we're storing them in a hash table, meaning that, for example, the color is the key in the hash table, and the value that the key returns are the locations where that certain color appears. For example, a hash table would return for green the locations, I don't know, this is 5 or 6 or whatever, would return the location here and also here when you look for the green color, and then for the blue color, it's something else, right? So it's just providing you the locations. And what we're doing is that we're doing the same for the read. And then what we're really doing is that we're querying the hash table with the colors that we see from the read. And then why we are doing that? So rather than applying a brute force approach for each character, what we can really do is that we can use the hash table so that we can perform a single lookup, instant lookup from the hash table, so that we can get the information whether that certain color appears anywhere in the reference genome. And if it does, which is very quick to answer that, we just try to collect all this information and then answer the question whether this read really belongs to the region in the reference genome or not. And then how we do that is basically, for example, here, for that read, we have the colors of blue, green, and the red. And then we have the same sequence of colors here in the reference genome, blue, green, and red. Okay, and then we say, okay, like we have the same sequence, and it may be likely that this sequence may belong here. Once we answer that, which is easier to answer, then we do the computationally expensive algorithms to figure out each character level differences between this region and this region. So what we've done essentially is that we reduce the problem from the entire reference genome to a smaller region so that we can only focus on that region to perform the computationally expensive steps. But even with those heuristics, analysis is really bottlenecked in read mapping. So this step is usually referred as read mapping because you're really mapping your read to a reference genome. But analysis is bottlenecked in read mapping in genome analysis. And why? This is because a human a sequencing machine can sequence a human genome in about 2 days, like 48 human genomes can be sequenced in about 2 days, meaning that a single human genome can be sequenced in an hour, right? So, but this is bottlenecked in the computational side because as the reads are produced from the sequencing machine, we can only map one human genome in 32 CPU hours, right? So meaning that, so this problem requires like many cores. So, which means that this is, the problem is really bottlenecked over here. And then, for example, if it required like one human genome, one CPU hours, like probably we would only have like this, the fast streaming of the data to the cores, and then the analysis wouldn't be bottlenecked. And to solve this problem, we require many cores, but like, of course, this is an issue about scalability, right? So then our goal is basically to come up with the intelligent design for an intelligent genome analysis. And what I mean by intelligent genome analysis is we want to achieve high bandwidth so that we can perform fast genome analysis. So we want to improve the energy efficiency and the latency of the algorithms or the hardware that we're relying on, so that we can develop like really energy efficient architectures, so that we can solve the problems maybe on the site without requiring high computational powers. Also, the DNA itself is really valuable, right? It's literally providing secrets about your genome. And then we want to perform all these operations in a private manner. And then we want the algorithms to be scalable, so that if you have like many cores, we want to utilize all of them so that they can, so the algorithms should be scalable. And of course, all the analysis that you're doing should be accurate, right? If it is not accurate, it's almost meaningless, because like later on, what we're going to do is we're going to be applying those predictions, for example, on an individual, for example, to a human, meaning that if we really apply something incorrect, this may have a serious effects on that individual's life, right? So meaning it should be accurate so that individuals can feel safe about the DNA analysis, the genome analysis that we're doing, right? So if it is not safe, as you may remember from the Maslow's hierarchy, no one is going to really trust, no one is really going to rely on the genome analysis that we're doing here so that they can apply our predictions that we're doing. So accuracy is really important. So throughout this course, we're going to be covering really different flavors in this semester. For example, we're going to be talking about read mapping, and then we're going to be talking about the papers that we published. So these are the topics that we covered last semester. So we covered like many topics. We can add new topics based on like because like we have these recently published works, so we can also talk about them so that you can get to know the state of the art research that we're doing in our group. So, but again, like one of the goals is really to accelerate, for example, read mapping. Not just read mapping, basically it's really many, we have many steps in genome analysis, right? So as I mentioned, like we get the sample, we provide in a sequencing machine, we have the reads, and then we map them, but later we also try to predict the variations between the individuals. And even in a single step, which is read mapping, we have like these many lower level steps, which is, for example, how we store these small pieces in your DNA. How should we store? Like, should we use a hash table? Should we use graph? Should we use any other structure, right? And then how should we also process this data so that the later steps can be fast? So there are basically many heuristics that we're applying even in the read mapping step. And then you can imagine like how many other steps that we may have in all the other steps. And then all of these heuristics also come with the opportunities to accelerate them, right? So there are basically many opportunities to accelerate many steps in read mapping and also in all of these steps. And then hopefully we're going to be learning more about these steps in the future. And so, as I said, we publish many papers in this topic, so you can read about them. So I think these are all linked from the course website, so you can read them to learn more about the topics that we're going to cover, also to learn more about the state-of-the-art research that we're doing in our group, right? So that we also have videos we publish, or videos and lectures, not just on genome analysis, but also like on many topics on computer architecture, etc. So you can just visit our YouTube channel so that you can watch all of these, including this one currently, which is currently being live streamed on YouTube. And yeah, and we have basically 2 project and seminar genomics courses. One of them is mobile genomics, and another one is accelerating genomics, which is this one. And you can visit the websites from the previous semesters to see what we've covered in these courses. And yeah, you can see the links here. So these are the more lectures on genome analysis, and these are some research that we've published previously in our group. So I guess that's all for now for the intro part. Do you have any questions so far? Okay, I hope everything was clear. Right now, what I'm going to do is that I'm going to stop the live stream, and so that we can talk about the project. So the project part is not going to be a live stream. All right. Okay, then. I'm starting live stream now.